So a number of people have said this is not a dihydrogen cross because it doesn't segregate into a 9:3 to 3:1 ratio. Yeah, you're right. And someone wants to make that argument. That's fine. I'm not tremendously concerned about those specific details. So you make assignments because there's this logic you have to follow through. There's one right answer and this, that, and the other thing. I write questions. Are you getting it? Did you run through the thought process and did you struggle with concepts and clarify things in your mind? That's what I'm trying to accomplish, right? If you justify that it's a monohybrid cross, that sex determination doesn't segregate the same way, I can live with that, right? So, today, I don't understand this stuff, but when I was in school, you went to school and you stayed at school. My daughter's in kindergarten. She have what they call early release days. We're gonna give the kindergarten teacher an extra hour off today. Yeah, I remember those were great. They didn't exist back then. 1 hour was like good. They didn't have these things, right? I'm almost that old that I can do that, right? So I'm actually going to try to finish it about 1/4 till. That will allow me to run to my car and drive the speed limit to my daughter's kindergarten, pick her up and take her to the daycare and run back to teach mine too. So I have kind of looked over the material and I have modified this PowerPoint file and I will upload a modified PowerPoint file. I modified it by taking some slides out. Less information about that. We will cover that stuff in more detail when we get to the next section. The same way this one I started like, here's your monohybrid cross, and the ratio comes out this, and it's absolutely perfect. And then I started saying, well, you know, red and white, and you get red and white out. Oh, what if you get red and white and you get a pink? We talked about how you get these messy in-betweens, right? And I hopefully am muddying the waters enough that you go, okay, there's going to be exceptions to it. In the next section, we will start talking about gene regulation in the very simple ways that they learned in 30s or 50s. And the only ways that existed, the only ways people knew about when I started graduate school. And while I was in graduate school, they figured out all this other stuff. Oh my gosh, it gets so messy and so complicated. I will try to describe it. I'll show you a couple examples. And it's kind of like, if you can imagine that a gene would be regulated some certain way, it probably is. There's probably one out there that is. It's pretty amazing. So I want to go through what we were talking about first. And this is great because you might mix these up. So it's likely to be an exam question, right? We were talking about maternal inheritance last time. And maternal inheritance is most organelles are inherited through the egg. So organelle DNA, mitochondrial DNA, chloroplast DNA is inherited from the mother inheritance. And now we're talking about maternal effect. That is not maternal inheritance. Maternal impact on the embryo. And we were talking about this in the context of the spiral, the direction of the spiral, right hand or left hand, dextral or sinistral. And last time what I did is I said, okay, we have a pure breeding dextral and a pure breeding sinistral. We breed them together. We got dextral. We breed them together. And if it were a monohybrid cross, you'd expect a 3:1 ratio between dextral and sinistral, right? What I told you is you get all dextral. Okay, so this must be maternal inheritance. It must be an organelle that's doing it. No, that's not the case. Turns out, so this is the parent, P0. This is the F1. This is the F2. If we do an F3, if we self-cross these guys on each other, we actually get dextral to sinistral in a 3:1 ratio. If we were to reverse it and we had the sinistral by dextral, we get sinistral, sinistral, and we get sinistral to dextral 3:1 ratio. So the way you can look at this to determine if it's a maternal effect, basically what's happening is these guys are genotypically if you to characterize the DNA sequence, these guys genotypically are 3 to 1. But you don't see what genes they have because this generation told them what to be. And the reason these guys are 3 to 1, genotypically these guys are not a 3 to 1 ratio. If you sequence the DNA, you'll see what they are. The reason these guys are the 3 to 1 ratio is because the 3 to 1 ratio of the parent generation, their DNA said you will match ours, right? So, this is the way it works. The cells, the mom's cells around the egg, tell the egg whether it should spiral this way or that way. That's why the genotype of the egg doesn't matter. That's why the 3 to 1 ratio isn't seen here, it's seen a generation later. The egg here wants to do something different, but the mother cells in this 3 to 1 ratio are telling it to spiral this way or that way. Does that make sense? Okay. Maternal inheritance, organelle. Maternal effect is the mother telling the next generation what to be. Okay. Questions on it? Yeah.
For humans, like, do they have examples for those?
Are there examples for that in humans? Not off, I don't know off the top of my head. I'll bet you somebody somewhere has found one. Okay. Okay, now we're going to talk about epigenetic inheritance. Wow, that's a terrible slide. In graduate school, I learned you never ever put a slide of text on. And there would be people giving their PhD thesis. They go, hi, I'm so-and-so, and they put a slide up, and it's all text. And you could actually see and hear everyone in class would slip down in the chairs and open books up or whatever, totally tune out. You never ever do that. You always have a picture. In biology, you can do it. Physics, maybe you can't do it. But in biology, you always have a picture. So, sorry about that. So let's talk about epigenetics. Epi means outside, right? In genetics, so it's outside genetic inheritance. So these are things that are not inherited. You express your phenotype on genetics. The phenotype you express is not necessarily correlated to the sequence that you inherit. Okay? You genotypically may be one thing, but you look different. And just because you look different doesn't mean you change the sequence of your DNA. The next generation could come out and genotypically be this and look different than you. Yeah.
So it's not like a tolerance gene, but not enough nutrients in the environment. So it's really short.
No, I would call that more of an environmental effect. So,
All right, so this isn't quite environment.
No.
Sort of.
For today,
Right.
it's not environment. The question for those of you on the camera. So if you're going to be tall, but you have poor nutrients and you grow to be short, is that it? No, and I'd say that's environmental. Although environment can affect this, and we get into this at the tail end of the next section of the course in gene regulation. Okay. So there are a couple of examples we can run through here to show you this. The first is dosage compensation. So, wow, I'm sorry that this is so light. People on camera probably weren't able to see it. I need to keep fresh markers in my stock here. So, to scale, these are about the size of the chromosomes. The X chromosomes are normal, full-size chromosomes. The Y chromosome is this tiny little nub of a chromosome. Not many genes on it, just a few genes. X chromosome has a lot, I don't know, 100 genes on it, I don't know. So it turns out, we could pick, and I'll give you the specific example of Drosophila. There is a variant of Drosophila where the eye color is called apricot. And a fly that inherits the apricot allele, that apricot allele is carried on the X chromosome. The other version of it is probably a brick red. I don't know the details on this. Okay? So it turns out, in the females here, and this goes for humans too, if there's some gene on the X chromosome, and there's a transcription factor floating around, you're going to express that gene. That transcription factor is going to bind here. And then another version of that protein will bind here. And so off of 2 different chromosomes, that gene will be expressed, which means there'll be twice the amount of RNA that can be produced off of this guy, right? So if this guy has a transcription factor that's going to turn on that gene, you've got 2 different templates making RNA. And here you've only got 1 template making RNA. You get double the RNA, right? So you get double the amount of protein. It actually, it's more exponential than that. Because each one, you have an amplification here. So you get 100 copies here and you get 1,000 copies here of the protein. So you say, okay, so maybe what we do is on the Y chromosome we have things that define maleness, and on the X chromosome we have things that define femaleness. And that's fine. You just express a bunch of femaleness stuff here, and you don't express much femaleness stuff here, so that the male stuff kind of overrides it, maybe. That's not the way it works, because an eye color gene doesn't matter if it's male or female. There are genes on here that are not related to being male or female. And there are like metabolic genes, like in glycolysis. So we have this thing called dosage compensation. And there are a couple of ways to dosage compensate. If we've got 1,000 proteins here, and this system gives us 100 proteins over here, how can we dosage compensate so that whether it's male or female, it's going to have the same number of glucose-6 hexokinase proteins made?
Inactivate the X chromosome.
We could inactivate this guy. We could go in with a laser shooting somebody and destroy this. So now we can only make this, and we'll only make 100 proteins off of that, right? Okay, what's another solution? Eliminate expression from 1X in females. What's the other possibility?
Increase expression in the
Double expression in males. I'm going to say with just 1X. Okay? So different organisms use different methods to get what we call dosage compensation, to get equal expression from the X in a female. The X is in a female gets the same expression intensity as expression in a male. Does that make sense? So, turns out, is this guy, for the X chromosome, is this organism homozygous or heterozygous? It's hemizygous, right? Because it's up there. Right? The correct term for this is hemizygous for the X chromosome. So in these guys, they express the apricot gene. They have apricot-colored eyes. And it turns out over here, how does that work out? I'm trying to run now. They have lighter color because, oh yeah, no. So, gosh, no, I'm losing my, oh yeah. So these are apricot colored. No, I'm losing this. I used to know this pattern really easy. these guys increase expression. That ought to be the same, right? So because they have a single copy on the X, their eyes are apricot color. Females homozygous for the apricot allele have apricot colored eyes. So probably what happens, sorry about this, is they decrease expression of this. I guess I have to go to the next slide. They kind of get rid of this. The effect is they're going to express less. Oh, no, I forget. Sorry about this. Oh yeah, okay, so females that are apricot, like this. That's it, yeah. So those females have apricot colored eyes, just like this guy. So they look just like this guy. If a female has an apricot allele and a mutant allele, which is white, they'll be light apricot in color. Does that make sense? So there's a dosage compensation that occurs. So these guys, the heterozygote, this dosage compensation. Okay. So one copy of females is not the same as one copy of males. Because males either increase or females decrease if they have two copies, right? There are different ways of doing this. Sienna elegans, females, or they're actually hermaphrodites, are XX. Males are X and they don't have the other one. They don't have another chromosome. There's no Y for them. Isn't that funny? So what they do is they decrease expression off of one. Let's talk about in mammals, the relevant, how this happens. In mammals, there's this thing called bar body. How many of you have heard of bar body before? Most of you. So this is something that was discovered in microscopy. Is they would look in a microscope, they would have a cell, and they would stain all the DNA of the cell. This looks to me like it's been stained with lithium bromide. Where have you heard that word before, lithium bromide? Yeah, in lab. It's the stain for DNA that we use in lab that causes cancer. Anyway, it binds to DNA. We'll talk about how it does it later in section 4 of the course if we do it. There is this very densely packed bit of DNA in females only, and that is located on one side of the nucleus. So if you were to look at the nucleus, most of the DNA is kind of, a little diffusely all around, there's DNA everywhere here. And then there's always, in females, always this dense blob of DNA stuck on the side of the nucleus. What the heck is it? Well, someone is actually eventually able to take it out and purify that thing away, and they figured out it's an X chromosome. And basically what happens in human females is one of the X chromosomes just gets bundled up and knotted into this highly supercoiled DNA. It's just incredibly packed densely together. So much so that if your normal DNA is kind of flapping around like this, transcription factor can come floating along and find some spot and bind to its promoter and express the gene. If a chromosome is all packed and dense and tight like that, say that line is as long as that line, there's no way the transcription factor is going to come in to find its promoter and bind to it and initiate expression off of that gene. DNA is hidden when it's packed up like that. It turns out the bar body in human females is like this, such that there is a single gene. There is always a loop of DNA with one gene poking off of it. We're going to call this X-ist. And in a bar body, that's the only gene that's expressed on the compacted chromosome.
Does that X-ist change ever? Is it the same gene?
It's always the same gene.
Always the same gene, they were changing between all
Yes.
individuals.
All individuals and all cells in one individual, female. That gene appears to be involved in compacting that chromosome, maintaining that chromosome as a compacted state. So this is the explanation for Calico Cats. So Calico Cats, there is a color gene on the X chromosome. And the color codes for a pigment which is either orange or brown, or black, I guess it is. One allele is black and one allele is orange. And Calico Cats have blotches on them that are either orange or black. Their underside is white, and there's a different set of genes that controls that. It's on their back. So I just had a calico cat. She passed away. But the way she grew, the way she developed in her embryo, at one point, so the limbs kind of develop out of the chest like this. And at one point, the limbs were basically connected. And there was a cell in this area that chose whether it was going to be orange or black. And that one cell on her chest chose to be black. And all cells that arose out of that were black. And that particular cell took up a little patch right here, and then out of that developed the limbs when she was in the uterus of her mother. Such that the limbs formed and half of the black dot, you could see, it was like half a circle, was on this limb, and the other half was over here. If you took her arms and laid her on, you got this perfect circle there. So what happens in mammalian females is when the embryo is at some early stage, all of the cells have two X's, and both X's are working. And very, very early in embryogenesis, every cell in the female, we think, randomly chooses one of the two X's to compact in a bar body and never express the genes off of there. And the other chromosome hangs around. So my cat, Ellie the cat was a heterozygote. She had one orange allele and she had one black allele. In some cells, the orange allele got stuffed away and was never expressed again. In other cells, the orange allele was stuffed away and never expressed again. Right? So she wound up getting these patches. Now every cell, if this cell here chooses to be orange, when that cell divides, every cell that arises out of that will be orange in color. And if this cell over here chooses to be black, every cell resulting from a cell division rising out of that will be black. And so that's why you get the patches. Does that make sense? Yeah.
Do males don't have that?
No, because males only have one X. I had a student 6 years ago who had a male calico cat. It's impossible, right? That was a male calico cat that inherited 2 X chromosomes and 1 Y. If you did a karyotype, it had an extra X chromosome, such that the 2 X chromosome said, oh, one of us has to inactivate into a body. Gesundheit.
I think he was lying.
She was lying to me.
She was
There are documented cases of it. There are humans who are XXY.
Just weird.
It happens. If you look in the literature, there's the first ones that came out, they're like, oh my god, I have an XXY cat. And then you're like, yeah, I have 20 over here. I gave away my example. That's a great example for an exam. Wow. You guys have questions on the bar body?
When, this is not about my body, but like, let's say there's a male with an XXY and he has a child, like, will he always just give that Y? Or will that not?
So, yeah, so let's do a Punnett square and work that out. That's a great question. The question is, if you have a male that's XXY, and that male has a child, what are the possibilities? So, the female, hopefully, is going to offer either an X or an X, say X1 or X2. And the male is going to offer an X or an XX, or an Y, or an XY. Those are the possible outcomes from this cross. And this happens. There are XY males that go on and have kids. That's it.
So does that triple X have 2 bar bodies then?
Yes. Now there seems, when there's an XXX mammal, it seems like the second bar body doesn't quite fully form. So there are some genes that are expressed off of it. And in text, they're described as women who are XXX appear to be very female-y, like you would see on a famous swimsuit cover or something, such that XXX is more common in certain industries or film type things. Got it. Got it? You understand what I'm talking about?
Have there been cases of where you have 2 X's but the proteins are dysfunctional, so you don't have bar body?
My guess is, so the question is there ever been a case where there's a human where a human female, 2 X's, but a bar body doesn't form in the cells? My guess is that would be lethal, but I don't know. I've not looked at that. Okay.
Can there be like a quadruple X ever or anything? Or does it only go to 3? Like if an XXX was mated with an XXY?
Yeah. I think I used to know the answer to this. I don't. I think that someone who survives that has serious
Illnesses.
yeah, beyond the very female-y kind of thing.
Oh.
It has a lot of physical disfigurements and mental
Okay.
challenges. I don't know. I mean, these are things you can go online. Okay. So, there is a gene that many of you probably know about. This is, I think it's Glucose-6-Phosphate Dehydrogenase, right? That's in glycolysis. I think that's Glucose-6-Phosphate Dehydrogenase. There are 2 different alleles in the human population. There is one where there's an RNA. Both of these RNAs are made. One of these, so we have our ATG, same thing. The sequence is almost identical, and here we have our TGA. But over in this guy, here's the TGA, but there is one nucleotide that's different in this allele compared to this allele, such that there is an earlier termination codon. So that the protein made off of here is this big, and the protein made off of here is this big. It's smaller. There are less amino acids in the protein. This exists for real in the human population. So if you have a female who is a homozygous, has one full length and one shortened allele of this gene, if you were to take a bunch of cells from the female, say cells out of the blood, you'll get a mix of cells. Some cells that have this version active and makes a protein this long, and some cells will have a shorter version of the protein. In that huge population, you'll get both types of proteins. You can run those out on a gel, and just like DNA, small proteins run faster than large proteins, and you wind up with 2 bands. That's the small allele, and that's a large allele. If you go to a female and you say, I'm going to take a single cell off of the skin of this female, that's heterozygote, and I'm going to put that in a dish and grow it all up, all of the cells are clonal. All of those cells came from that one original cell that had chosen which was the bar body. So all of the cells in the dish will express only one form of the protein, either long or short. And then you could go like to the liver of that female and take a different cell, and maybe that cell has chosen a different X to be the bar body. such that you could grow a clonal group up and you could get cells that have only one band or the other band. So there are, 100 genes on the human chromosome, X. And every gene on the human chromosome has different alleles. So the human females in this room, you are mosaics. Part of you are expressing certain X genes, certain versions of your X genes in some parts of your body. In other parts of your body, you have patches that are expressing other alleles of your X chromosome. This manifests, there's one common one in females. Sometimes there will be women that have soft, peachy body hair. Then they'll have patches where they've got this thick black tip, stiff body hair, and just like a patch like right here. They're calico women because they have different alleles for hair thickness and hair color. Every one of you, females in this room, is calico in some characteristics. How cool is that? You're versatile, right?
Ever seen Cats the musical?
Cats the musical? Yeah, that's totally off topic.
It's kitty cat.
Oh, it has a calico cat, great. Okay, there we go.
I mean, it's a person dressed up as a cat.
Okay, so this is the experiment where they just showed what I showed there. Okay. Is that really cool or what? I think that's awesome. Okay. Yeah, so in this region, at the time this is written, I actually have a little more detail than this in what's been uploaded online. Just understand that there's one region on the X chromosome where there's a single gene that's going to be expressed. It's out of the, XIC region is this loop that's exposed. We talk about how the Barr body forms later on in the semester. Okay? The process for how this happens has been pretty well described. What happens is when there's the original X, if this cell or this X is going to become the Barr body, what happens is the region around the X, what is it, CIX, is that what it was? XIST gene, basically. The region around that gets compacted in densely compacted DNA, and then you can actually watch in a microscope as the compaction kind of tightens up all around and just kind of pulls all this stuff in. And eventually the whole thing gets all tight and packed except for that one little loop. So the DNA gets tightly compacted. The mechanism of this involves covalent modification. Functional groups put physically on the DNA, and functional groups put on the histones that are associated with the DNA. And this is called the histone code, and this is the basis of epigenetic inheritance. And it comes together and ties into gene regulation, which we'll talk about in the next section of the course. At this point, just understand that it exists. DNA can be covalently modified to regulate it. This is also the explanation for what people call genomic imprinting. You could inherit the brown-eyed allele and the brown-eyed allele from your mom, brown-eyed allele from your mom and brown-eyed allele from your dad. And, well, let's see, if you got blue from your dad and brown from your mom, the other way around, yeah, from your mom, I guess it could go either way, it doesn't matter. You can inherit like a blue-eyed allele from your mom and a brown-eyed allele from your dad, and you say, okay, well, I'm heterozygote, so I should be brown-eyed. But in your father, the father can chemically modify the DNA such that the transcription factor can't find the brown allele. The only allele you can then express is the blue from your mom. So even though you have genetically you should be brown-eyed, you can be blue-eyed because covalent modification pieces of the DNA that happen during meiosis. This stuff is, the patterns of how this happens are so complicated. In the next section, we'll talk about a little bit about it, but we won't get into excruciating detail. Okay. The functional groups, generally we think of being put on the DNA or methyl groups that can put on the DNA. Okay. So that's what I've got. I covered that last little bit, just skirting over the surface, introduce you to the idea of bar bodies. We'll come back and explain how that all happens later. Did you guys have questions on this? We're going to have a review session Friday, right? Exam on Monday. I guess I need to get writing an exam. That might help. That helps.
Helped.
Any questions at this point?
So is what's online not updated?
No, I updated on my computer and then I came down with the original crap and fixed that file. I have to load that one up. That has to happen after my drive too, so an hour and a half from now at earliest.
Okay.
Okay? Mosaics and gentlemen? Calicos and gentlemen? All right. If you haven't turned in your homework assignment, do that. So right here.